All right, everybody, it's time to start moving to Test 2 material for our Bio 207 hybrid class here at St. Louis Community College. This Unit 2, Test 2 lecture material will be over bones and joints. So let's dig into Chapter 6 and our osseous tissue, which is another name for our bone tissue. All right. When we start talking about the organ system of the skeletal muscle or skeletal system, what we're talking about is physically the bones that make up our body. And then we also need to talk about the cartilage, the bone marrow that's encased in the epiphysis of our long bones and our flat bone, spongy bone area that are going to make our red blood cells, white blood cells, and platelets. We also need to talk about the bones and cartilage providing protection and the leverage and the ability for physics of making joints move that we incur with this system. So there's a lot going on with the bones. However, we tend to only really focus on the anatomy, and we sometimes neglect all of the physiology that the bones are providing for us in our bodies as well. So try not to forget that, yes, it is living tissue, and it does adapt and change as the environmental and physical cues on our bodies are changing. So if we go to space, that's one of the big things about potentially going to Mars is how can we maintain bone mass at a sufficient amount that when we get to Mars, we won't potentially try to move on the planet under a little bit of gravity and our bones will all break and crack. Or how do we get back to Earth and not have potentially long-lasting problems from the bone loss that occurs under microgravity conditions? Okay, so there's a lot to the system that we sometimes forget about because we're worried about just naming the bones. All right. When we look at how do we start classifying bones, again, that is the anatomy part, and it is an important part, especially if you are desiring to go into a field of physical therapy, occupational therapy, exercise science, and then you have to learn how to, again, do the physics of the moment angles that the bones and the joints are creating. And you're going to learn about, again, the musculoskeletal system and improving that system. So bones, from an anatomy standpoint, when they looked at skeletons, how did they divide them out initially into categories? And the first way to categorize the bones was to look at the shapes and kind of put bones of similar shapes together. So when we look at the 6 categories of bones divided out by shape, we see that we are going to get, some groups are bigger than others, but most of the long bones get thrown together, so then the short bones, and then our flat bones, and then some of them kind of fall into this weird category where they are oddly shaped, and so we kind of throw them all together as irregular looking bones. The 2 categories of bones that are listed there that are very small in number are going to be our sutural bones, which we're going to see as the fontanelles of our baby head is slowly converting into bone and those skull bones are forming together. There is potentially little fragments of bone that are not part of any major frontal bone, parietal bone, and it's embedded in the suture. And so those little independent bones, we're going to call those the suture bones. For the sesamoid bones, the one that comes to mind the easiest for most people to recognize is your patella in your knee joint. It's very easily the biggest of the sesamoid bones, and it forms in between these tendons. But you'll have other sesamoid bones maybe form during your lifetime for a variety of reasons. But again, most of the time the patella is the one that we normally think of. Okay. Now, beyond just those structural categories, we do also look at how bones are internally organized. And so if the bone has a predominantly large amount of trabecular or spongy bone, we tend to group those bones together. And then if the bone is going to be like our long bones, predominantly large areas of compact bone, we'll kind of group those together. And then our last way of classifying bones is we tend to look at some of the unique features that they contain and take that into account as we put them into categories. Okay, so for the classification of bones by shape, again, sutural bones, they would be bone fragments that are formed as dermal ossification is happening between the skull bones. Again, your fontanelles, ultimately, when you're a baby, have to go away, and your skull has to merge and fuse, and the fusion joints here are sutural joints. Well, technically speaking, some little bone fragments can form independent of those other bones. And so we're not going to normally give them any other special name other than sutural bones, and to see them in most of our models is non-existent. I mean, and in any case, we'd have to potentially look under a microscope for these bones to be found in between the fused looking sutures of the skull. Okay. The next category, irregular bones. Your vertebrae fall into this category. So there's pieces of vertebrae that are kind of pointy. Some of them are small, some of them start to get big. But for the most part, it's a oddly shaped bone. And so because of that odd shape, we throw it in here with an irregular bone category. For the short bones, we tend to see them small and thick. So the wrist bones, the foot bones are great category, great examples of this, whereas the long bones we are going to see are most of our limbs, so the bones that are rectangular in nature and size. And then our flat bones, we're going to have areas where we sacrifice width, so that way there's a little bit of compact bone sandwiching some spongy or trabecular bone and red bone marrow. And so we're trying to get something maybe light in this situation, so that way we are protecting underlying tissue, but we're doing it without having a lot of weight added on top of it. And again, our sesamoid bones develop in tendons, in ligaments, and again, we usually only think of a very large pronounced one example, and that's the patella, right? I don't know why this is going so fast. So looking at the third category of how we start to label bones is we start looking at bone markings. All right, we start looking for depressions and grooves and holes in the bones. And so what we're going to find is there's this whole terminology of what all of these markings are. Usually some type of mound or outward spike or hill on the bone is a projection. And then any kind of inward kind of sinking of our valley is some type of is usually some type of depression. And so depending upon the bone, we will find that sometimes we always name the projections a certain terminology on a certain bone, and we never use that terminology again. So I think of like the femur. The only place we have the greater and lesser trochanter to define those projections on the bones is on the femur. Otherwise, we use different categories and terms. So the takeaway is from your book, Table 5.1 or 6.1, and in the older version of the book, Table 6.1 was organized as shown over here, you need to learn these terms. And so expect these terms as a matching type question on your lecture exam. Expect these terms as questions that will be asked on the lab exam. The second reason why you want to learn them is in some ways it'll help you with your bone identification. So yes, we're going to expect you to learn the names of all the bones of the body, and luckily most of them are paired, so you know, very quickly 206 bones becomes about 103 because 2 bones exist, the right one and the left one. But the other thing is, once you can know the bone name, and then it becomes a little easier to differentiate when to use trochanter versus tuberosity. And so you can then maybe help yourself do a little bit better on that lab exam by making sure you learn your bone, and then for each bone, maybe what are some of the unique terms for these projections or depressions that are incorporated. All right? So that's what this slide is trying to tell you to do. Great way to do this is to make flashcards and practice, practice, practice. All right, so moving into long bones. We have quite a few of the long bones of our arms and legs, and the long bones are important for movement, so important for leverage, and the long bones have a little bit of a uniformness to them when we talk about the internal structures. So we need to kind of break down long bones into their anatomical features that are going to, for the most part, show up in every long bone. All right, so the picture here depicts a femur, but if we threw a humerus, a radius, a tibia, most of these are going to have the same features located in these certain areas of the long bone. All right, so the long bone is kind of rectangular in shape. Its length is more so than its width. And because of that, we are going to see that towards the knobs or the ends, the superior, inferior, or the distal proximal part of the bone, there is normally going to be some type of enlargement and special features that show up preparing to articulate or form a joint with the surrounding bones that are next in line to it. And so that enlargement, that area of the distal proximal part of the long bone that gets ready to fit in a certain way with other bones and form joints is going to be the epiphysis of the long bones. And typically that area is going to have some cartilage around it, so that it'll have cartilage in the perichondrium, then it'll have a periosteum, and then it will have some compact bone, and then most of the underlying area is going to be spongy or trabecular bone. And the spongy or trabecular bone in the epiphysis is going to be filling in that hollow area, that hollow space with red bone marrow, which contains our hematopoietic stem cells, our stem cells that give rise to all of our blood cell elements. Okay? So we're looking at the epiphysis are going to be the ends of the long bone, and we are going to see that then the center, which tends to have a larger amount of compact bone here, is going to be our diaphysis. So epiphysis, epi for towards the ends, around. Diaphysis is going to be the center. Okay, a little bit of an imaginary area where the two bones kind of come together, the epiphysis and the diaphysis, will be our metaphysis. So that's that middle kind of joining spot. All right, again, the epiphysis tend to be larger in red bone marrow because of the spongy, larger accommodations of spongy bone. The center of the diaphysis is going to have an open cavity that's known as the medullary cavity, and it is going to be more of yellow bone marrow. And yellow bone marrow is adipose cell rich, and it's not there to give rise to what will become most of your bone cells. It's there to cushion, support, insulate, and store fuel. All right? The bone will have blood supply, so coming into the periosteum will be some arteries and veins. They will come into that periosteum, and then because of the way that spongy and trabecular bone are, they have to eventually get to and spread out in that red bone marrow area, and then we'll also see major blood vessels that push through the periosteum, push through some of the perforating canals, and get into the medullary cavity as well. And then small capillaries will be running, again, through our osteons, through our central canals, and other perforating canals, providing smaller vessel level support to the trapped bone cells living inside our functional unit, which is our osteon of compact bone. Okay, so remember from last tissues, peri, around, bone, this is connective tissue. That connective tissue that has a very strong collagen-rich outer part that allows for the collagen fibers to interweave with the ligaments and tendons. So that is how the connection is made of muscle attaching to the bone, is through their connective tissue interacting and weaving and forming this strong connection to the periosteum. And this is how ligaments connect two bones together as well. An endoosteum is going to be in that medullary cavity. Technically, it's kind of an open space. So it's going to be lined by very similar kind of protective connective tissue. And so that connective tissue there, because it's inside, is an endoosteum versus, again, peri usually means outside and around. Right? So those are some of the key terms we need to focus on related to long bone features, and those are kind of going to transcend and be found through all of our long bones. All right? When we look at our flat bones, some unique kind of cohesiveness occurs there as well. Typically, flat bones are going to be more about the width and maybe not so much the height or the length. And there is compact bone towards the margin, so it's kind of sort of like a sandwich. The breading is the compact bone, so there are osteons, there are some Habersham canals or central canals, perforating canals, trapped osteon cells, concentric, interstitial, lamellae, but the majority of the bone mass is going to be spongy, calcaneous, trabecular bone, which we see in the center, that somewhat like the wiring of bridges allows for the load and the stress application to be handled, but allows the bone to maintain somewhat a lightness. And then that lightness and that hollow open space that is created allows for more red bone marrow to take up residence, so we also have more hematopoietic stem cells available in our body to give rise to our blood cells of our circulating in our blood. All right. So we see that compact and flat bones, the skull bones, the hip bones, part of the scapula, the sternum or the breastbone. And when it comes time to get bone marrow for, trying to help with kids get new cells, new red blood cells, help them as a treatment for leukemias, we take donations from people typically out of the hip because it's a little bit easier to suction out some of that bone marrow and get those stem cells that way than it is to try to, with a needle, with a drill, punch into the skull or the breastbone. And again, small margin of error. If you miss and go too deep, you could be in brain tissue if you try to go through the skull or get the skull, or you could end up in the lungs or the heart if you miss with the breastbone. So, it's kind of just easier to talk about getting bone marrow out of the hip. Again, when we look at the compact spongy bone specific to flat bones, it's also sometimes known as diploid bone. Okay? So let's again review from that very microscopic view, compact bone. All right? Compact bone is going to have osteocytes. Now, osteocytes collectively is a term for all of the cells within the bone system. But those cells can be further differentiated into osteoblasts, which are cells that are adding to the matrix by putting down collagen, putting down calcium phosphate crystals. You have osteoclasts, which are cleanup cells. That's how I remember it. The clast, the C, is a cleanup cell. This is a cell that's taking away matrix, dissolving it, liberating that calcium to potentially go into our blood supply. Right? And the blast and the clast are constantly kind of opposing each other. As one is building, the other is taking away. And so in doing that, that's how we remodel and reshape our skeleton, especially when we're loading it with weight-bearing activities like you do in the gym to build stronger bones. Right? The other cell population is a progenitor cell. It's known as an osteoprogenitor cell. And so we refer to these cells as having the potential to be converted into osteoblasts. Now, any cell that gets trapped and becomes a resident cell stuck in its position using canaliculi, those little digits, those grooves, and extends its little cellular projections towards the bloodstream that's in the central canal or perforating canals, those you can just refer to again as an osteocyte. And so you'll see osteocyte right there. Okay? The picture here is showing you potentially both features of, here is compact bone with those central canals carrying blood vessels up and down the length of the bone, and it is surrounded by circular matrix with trapped cells within those kind of circular matrix pieces, and that is known as an osteon. And in the osteon, which is that functional unit, each of these little circles around the canal is known as your concentric lamellae. Okay, and lamellae is another term for the matrix. And the matrix, again, is calcium in its crystallized form, so it binds to carbonate and phosphate and different chemicals, ions, and then collagen proteins. And collagen protein is important because collagen gives a little bit of flexibility to the bone, so as we're pulling on it with our muscles or our ligaments, the bone isn't brittle and doesn't completely fracture or break. Okay. Now, between the osteons, remember, you are going to have little space here and there that has to be kind of filled in with more matrix and trapped cells, and that's where our interstitial lamellae is existing. All right. And so the interstitial lamellae, if you had a bunch of circular pillars pushed together, there's still little cracks in space, and it's filling up that cracks in space, so therefore that area of bone is very dense, full of solid material. Wrapping around all of these logs and crack-filling material are going to be what's known as your circumferential lamellae. And so these are the circles that surround the, almost run the entire circular around the entire bone, okay, especially in our long bones. And they are going to be the ones, the circumferential lamellae, that is closest to the periosteum and the cells that reside in the innermost layer of the periosteum. And some of the cells in that innermost layer of the periosteum are some of those osteoprogenitor cells, osteoclast, and osteoblast. And so when I need to, when I am carrying lots of weight and I'm trying to push my bones to get stronger, get thicker, handle more weight-bearing load, I am triggering with that weight-bearing stress for my cells in my periosteum layer to become more osteoblasts, become more laying down matrix, adding to the circumferential lamellae. And so in this case, instead of having maybe 4 rings of lamellae around all of the compact osteons and interstitial lamellae, maybe I can get that to grow out to 6 or 7. Right? And so that's how, again, weight-bearing activity helps us increase our bone mass, because it helps us grow in a little bit of appositional width by adding circumferential lamellae. The outermost part of that periosteum, and you can see the blood vessels are sitting in there, more of your major blood vessels coming into and out of the bone. That is going to be more of a fibrous connective tissue, so a collagen-rich area that allows for somewhat braiding and interweaving and formation of the tendons and ligaments seamlessly kind of binding and becoming part of the collagen fibers within this layer. All right, so that is where all the connections are made, and so technically speaking, the muscle cells are never actually touching the osteocytes. It's the connective tissue around the muscle and the collagen fibers that are part of that connective tissue that connect into and form kind of braiding, chain links, and different types of patterns that then are part of the periosteum connective outer piece, okay? All right, moving towards, again, in the long bones, towards that medullary cavity, you see that the transition occurs where the compact bone gives rise to trabecular spongy bone, calcaneus bone. And when we look at the calcaneus spongy bone here, we're going to see it's very much like the interstitial. There's no really unified circular with a central blood vessel feature of an osteon. It's just the material forming these lines, these pillars, these columns that interconnect to each other at certain angles that are most efficient at bearing the load, but allow for a lot of open space. And so that's where in the medullary cavity of the long bone, we're going to see fat, rich bone marrow take up residence. So that spongy bone is going to represent not the epiphysis, not the flat bones, but the medullary cavity transition of bone into open space, and we're going to see bone marrow take up that air space. Okay. Technically speaking, the outermost part of all of these pillars of trabecular spongy bone are going to have a very similar type of connective tissue wrapping around it. And that connective tissue component is going to be your endosteum. And it's very similar to the periosteum in that there's cells that live in there that are going to have that potential to convert to osteoblasts or osteoclasts and remodel the trabecular bone of the medullary cavity, add to it in thickness and girth, or take it away. Okay, so that kind of gives us, for a long bone, talking of the, let's say the diaphysis, from the outermost superficial periosteum inside to the medullary cavity, we expect to see connective tissue related to a mix of osteoblast class, osteoprogenitor cells, and then fibroblasts and immune cells, and collagen-rich fiber area for the periosteum. Then we see the organization of your compact bone, so lots of osteons, filling gaps between the logs of the osteons, some interstitial lamellae, filling the space between all of those osteons and interstitial lamellae, and the periosteum being some circumferential lamellae that runs the complete circle route around, like the periosteum, around the entire long bone diaphysis. Transition to a spongy bone with some endoosteum membrane around the pillars that are forming the trabecular bone, and then an open space that allows, again, for blood vessels that have penetrated through the periosteum, through perforating canals. Some of them have branched into the vessels running through the central canals, but eventually running through the center medullary cavity of the bone. Okay. So compact bone characteristics, it's very thick. So there's not a lot of watery interstitial fluid in there. It's very much, the matrix is hard calciumized crystals and collagen protein interlocking, connecting, forming a dense material. The cells that are stuck in this lamellae, in this matrix, cannot physically move because the material is so thick, so hard, that there's not an ability for the cells to wiggle around much. So the cells then dig into the matrix, little canals, little ditches that allow for cellular projections, somewhat like little arms, to grow into those holes and reach towards the blood supply. So the cell is most likely to then get its oxygen, its glucose, its fat, and deliver its nitrogenous waste and carbon dioxide to the blood supply. Okay. Again, the bone cells, you can collectively call all bone cells osteocytes, but for a better definition, osteocytes are in compact bone what we specifically call the cells that are trapped in the lamellae. The osteocytes are trapped. They can't move, but they do reside in a little chamber-like feature known as a lacunae, just like cartilage lived in a lacunae. And those osteocytes are responsible for maintaining the matrix. So they do both build and cleanup duties. And they kind of make sure that the matrix and their immediate surrounding is maintained to the efficient level of handling whatever stress is applied to them on a consistent basis. Okay? So again, canaliculi are grooves in that matrix of material. The periosteum is our outermost connective tissue that allows the blood vessels to come close to bone and push into bone, as well as houses a large population of cells that are either builder cells, so osteoblasts, osteoclasts, cleanup cells, osteoprogenitor cells that can become potentially blast and then eventually maybe get trapped and become osteocytes. All right. In addition, it's still connective tissue that has collagen fibers, so there could be some fibroblasts, there can be some immune cells that live in there. All right. Other names for the dense part of the bone matrix is cortical bone, dense bone, cortical comes from, it's the outermost bone of flat bones, and dense bone because there's not a lot of, again, open space in the matrix area. Because of the way calcium and its other ions it's binding to combine into a solid mass, it takes on mostly a white and smooth appearance, so that's why bone looks that way. And approximately 805 of, again, total bone mass. So it's a lot of bone mass. It's very heavy. Okay. When we look at the lamellae, again, the extracellular matrix of bone, we expect that most of what is that extracellular matrix is calcium ions that have been forced to bind to other ions, making a crystal. Phosphate is a big negatively charged molecule that we maintain in our cells and in our body as a ability to make AMP, ADP, ATP, GMP, GDP, GTP, and again, phosphate bonds being broken and maintained are our best way for energy to be expended or housed in our system. So when we have excess phosphate, we don't want to lose it all to our urination or excretion methods. So by binding phosphate, which has a tendency to maintain a negative ionic charge, to calcium, which has a positive ionic charge, it gives us the ability to bind calcium to a negative molecule that is readily ionically forming that bond, and then it also helps us keep phosphate in reserve should we need more phosphate for more potential phosphate-phosphate bonds with ADP, GDP, UTP, all those other nucleic acids that help us form energy molecules in the cells. Now, phosphate is not the only place we like to bind calcium. We can bind calcium to, again, hydroxide, which is OH negative typically, so calcium is 2+, so two hydroxides can form a neutral ionic crystal molecule with calcium. And you see hydroxyapatite, which is a combination of phosphate and hydroxide together. We see that collagen protein is the most abundant protein in the system, and then other ions like sodium, magnesium can find its way into the bone matrix, again, sodium being a positively charged ion, potassium being a positively charged ion, magnesium being a positively charged ion, and then carbonate. So carbonate comes from carbon molecules, or in many cases our glucose, our glycerol, our background molecules for energy expenditure. So sometimes we'll also use them to help be storage molecules for these ions. Most of what compact bone is inorganic, meaning it's these ions and molecules that form that might have a carbon atom and might have a hydrogen atom, but they don't have usually both a carbon and a hydrogen atom. So anything like collagen that's a protein, proteins have typically a central carbon and have a carboxyl group which has a hydrogen and a carbon component. So those are considered organic molecules because they have both carbon and hydrogen atoms as part of their features. Okay. I've been through this already, that the bone cells are usually going to take on collectively, when you refer to every bone cell, osteocyte, but we can use osteocyte more specifically for bone cells that are trapped in lamellae. And by being trapped in lamellae, they also do the housekeeping of their surrounding area by keeping the matrix sufficiently supplied with material, or take the material out as we need to liberate more calcium or other materials out of the bone matrix. When we look at the periosteum and the endosteum, we see that's where our osteoblasts and our osteoclasts, and again, build for blast, clean for class. So osteoclasts, or the majority of those cells, are all about taking matrix and dissolving it and liberating it back to its most simple elements, so calcium, sodium, potassium, magnesium, and phosphate. Whereas blasts are taking those ions and forcing them into crystallized hard features that are then deposited into the matrix along with collagen protein. Our osteoprogenitor cell, again, is the cell that has that capability to become an osteoblast and build more matrix, as well as putting matrix around itself and getting trapped in a new circumferential type lamellae or a layer in the trabecular spongy bone, and so it would potentially turn into an osteocyte. Okay? Again, why do all these cells exist? As living tissue, it can constantly be changed and modified, and to be able to be changed and modified, cells are going to respond to different signals, stimuli, and cues and take away matrix or build matrix. And so that's really what the osteocyte's doing in that tissue. So for compact bone, again, the osteon is the central circular log-type feature that is surrounded with concentric lamellae and with trapped osteocytes that are protruding through the canaliculi, these projections to the blood supply. The central canal is hollowed out, so an artery and a vein typically can run the length of the osteon up and down the diaphysis of the long bone. Right? There will be connections for this central canal with other central canals, and to get the blood supply to connect to those other vessels, that's where our perforating, our canals that run perpendicular to the central canal are going to be located. Right? For spongy bone, again, we transition into, like the little cartoon over here shows you, there is not a central open space, but we kind of see interstitial and some circumferential lamellae kind of form this nice distinctive beam or this nice distinctive pillar. And the pillar and the beams connect into other pillars and beams at angles, somewhat like the Eiffel Tower's architectural feature. If you look at bridges, again, going over the Mississippi River, making a solid bridge of material, it would be very heavy, and that might be very challenging for the weight-bearing beams pushing into the ground at the depth of the river. So trying to make the material that is the bridge as light as possible, you do this feature of architecture where you try to make pillars form into each other such that they maximize weight-bearing capacity, but they are as skinny, as light as possible, so the entire weight of the structure isn't too much for the load-bearing foundational components. Right? So in the epiphysis, in the flat bones, we're going to see a large majority of the spongy bone. We also see it in the transition areas where in our long bone medullary cavities, the compact bone will transition to a little bit of trabecular bone, and then that feature will end into an open medullary space. The space created by the pillars pressing into each other is going to be important because we can fill that space with either red or yellow bone marrow. And red bone marrow is important because it contains the precursor stem cells that give rise to all of our red, white, and platelet-creating cells. Right? Yellow bone marrow is mostly about holding fat, insulation, and storage for your fat. So fat, again, when these bone cells need to make energy to be able to produce collagen, produce matrix material around them, is going to be the way that these cells can make ATP and then expend that energy as needed. Okay. All right, so just to summarize the microscopic features, everybody needs to be aware of the key terminology for compact bone. Compact bone being in the cortex of our flat bones, so kind of like the breading of a sandwich. It's the outermost part. It's also going to be very dense and thick in the diaphysis of the long bones. So all of the terminology you see here, you should be able to apply to your cortical as well as compact bone pictures and models. For spongy bone, very similar terms, but again, spongy bone is found in the epiphysis of long bones and in the center of flat bones. And so you should be comfortable with the terminology associated with it. Again, for our cells, we tend to see that the bone is going to have trapped mature osteocytes that maintain the matrix around them, but whether this pillar is going to get thicker and stronger is being done because the margin, the outside of it, where our osteoprogenitor and osteoblasts can form, and where our osteoclast can form, should we need to smaller or take away some of that material. Okay? The periosteum is a very important part of bone, even though it's not bone tissue, it's more connective tissue. And the reason why is this is how we bring fluids of blood, and so oxygen and glucose and fat and amino acids and calcium and phosphate ions into the vicinity of our resident osteocyte cells. This is how blood vessels exit the bone and take away the carbon dioxide and other waste products. This is how the bone physically connects to other features of the body, to include ligaments and tendons that allow it then to be manipulated and pulled on as joint angles change and as the muscles pull and use the bones for leverage. All right, so the innermost layer is the cell layer, and that cell layer is a mix of cells that are precursors to becoming bone cells, and so laying down matrix, getting trapped in the matrix, becoming part of now what is the dense material that is compact circumferential lamellae, as well as non-living material that is collagen-rich fibers, fibroblasts there, and immune cells that, again, are present to invaginate, weave into, form chain links, form knots that connect physically muscle tendons to the bone and ligaments into the bone. Okay. Again, the functions. Periosteum protects, so the osteocytes, the actual crystal calcium carbonate, calcium phosphate, calcium hydroxate is not ever actually touching other organs. It provides the route for nerves, lymphatics, and circulatory vessels into and out of the bone. And it helps in the repair process because it's where the progenitor, the blast, and the clast cells live primarily, because they don't get trapped in there. Once they're trapped, they're osteocytes. And so if there's a damage or a need to thicken up, that is where the thickening up, the remodeling can happen, or the repair process can happen in that periosteum. The endosteum is very similar to the periosteum in terms of it's a place where osteoprogenitors, osteoblasts, and osteoclasts live, but the difference is it's inside the medullary cavity and it's wrapping around more trabecular or spongy bone instead of compact bone. So when the additions are made more on the endosteum, you're making the endosteum increase the width of the trabecular bone pillars or decrease the width of the trabecular bone pillars. Still a protection layer with some collagen fibers that are going to be around and somewhat protect and separate out where the bone marrow that is living in the medullary cavity will reside versus where the cell layer and the bone matrix will be located. So for the most part, we've covered the microscopic features of spongy versus compact bone. We've gone into a macroscopic view of if I get a big pile of bones, how can I separate them into categories that best puts like bones together. And you have a lot of bone terminology you need to get comfortable with, specifically to projections and depressions and holes. What do we call those special features typically in bones? So lots of vocabulary, lots of things you need to make flashcards on, lots of things you need to review and get comfortable with for both your lab exam as well as your lecture exam. The next video that I plan to do is going to focus more on bone remodel, bone growth, and bone maintenance with nutritional, exercise, and aging factors coming into play. So come back for the next video, and again, this material for the Bio 207 classes here at Meramec, this is going to be Test 2 material, not Test 1 material. All right, have a good day.